speaker is Professor Richard Douglas of New Zealand, who has been doing tremendous work on the microbiome in CRS, and will tell us all the important things we need to know about it and maybe comment on possible clinical applications. Richard, thanks very much for being with us, and we can't wait to listen to your talk.
Tim, thank you very much for the invitation to join you all. I nearly said this evening, because it's this evening, it's 11:30 here in New Zealand on Friday night, this afternoon for you in Athens. Now, I would like to screen share my Zoom meeting. Here we are. I would like to follow on from Peter's overview of the pathophysiology to focus much more specifically on the role of the nasal microbiome. And I'll begin by showing a scanning electron microscope image of the rather beautiful cilia which lie in the epithelium and maintain mucosal health by enabling the mucociliary blanket to function. And here's how an SEM looks like in a patient with chronic rhinosinusitis. And you can see that the cilia have been devastated. And here, with the eye of faith, it's possible to see some cocci on this devastated sinonasal mucosa. And the question that I'd really like to address is, how much of a pathogenic role does the microbial ecosystem of the nose play in the pathogenesis of this condition? Why is this important? It's so important because we prescribe so many antibiotics for the management of this condition. So the questions I like to address are, what is the microbiome? Secondly, is it different in patients with chronic rhinosinusitis compared to normal controls? Thirdly, is it affected by antibiotics and by surgery? Is it stable over time? And finally, does it have a plausible role in the pathogenesis of CRS? So to address the first question, what is the microbiome? Well, there are an awful lot of microbes in our nose. There are bacteria. There are archaea, or cyanobacteria, viruses. We all know that they certainly cause rhinitis and sinusitis, and the fungi. What I'd like to do for this brief talk is actually focus on only one of those groups, and that is the role of bacteria, because I think the pathogenic role for bacteria is perhaps better worked out than it is for those other groups. So how do you define the sinonasal microbiome? It is the combined genetic material of the bacterial microorganisms in a particular environment. Why is it important? Because the zoo of bacteria which grow on our mucosal surfaces present a significant barrier to potential pathogens which need to outcompete them in order to establish themselves on the mucosal surfaces. And secondly, the presence of that zoo of bacteria modulates a mucosal immune response. The microbiome has a specific structure. There are a whole lot of different niches on, within, below the sinonasal epithelium, which bacteria can grow in. And that we can have planktonic bacteria in the mucus, biofilms adherent to the surface, cells growing within the epithelium, and bacterial cells within the interstitium. And how you sample, how you measure, determine the microbiome will be influenced by what specific sample that you take, for example, a flocked nasal swab, which will predominantly take mucus specimens, will be different from, say, a cider brush, which will take biofilms, which may potentially be different from the culture of a biopsy or the determination of the microbiome of a biopsy, simply because you are sampling different niches. Once you've got your sample, how then do you actually physically determine the microbiome? Well, for most studies, it involves an amplification of a particular part of the chromosome or the DNA of the bacterial cell. It's the 16S part of the DNA, which is a highly preserved part of the DNA, which can be amplified and sequenced. And once you've got the sequence of the particular 16S portion of the DNA of the bacteria, you can then compare it to really vast databases, and you can see what sort of match you get. And in that way, you can determine the composition of the bacteria in your specimen. So the second question, is it microbiome different in patients with CRS compared to normal controls? And it's a fundamental question to ask. We asked this a few years ago when we put together a meta-analysis. All of the online data available from CRS microbiome studies, and we analyze them all through a single pipeline in order to reduce the variability between the studies. And we produced this graph, which is complicated, but if I can say that graph has the microbiomes of 131 different patients. But if we look at the microbiome of just 2 examples, the composition, you can see that X percentage of that particular patient were made up with actinobacteria, and that percentage of proteobacteria, and the percentage composition for the second patient was slightly different. And so when you put them all together and line them up according to, in this particular presentation, how much actinobacteria each specimen has from the lowest to the highest amount, hence a little green hill, you can see that there's an enormous inter-individual variation. And if you compare those data with the data from the control group, that the actual overall groups look actually pretty similar. And so we drew from this study 2 conclusions. One, that there is an enormous inter-individual variation in the composition of the microbiome from one CRS patient to the other. And secondly, that there was no really glaringly obvious consistent difference between the overall composition in CRS patients and controls. And we've recently repeated this meta-analysis. We're just doing the analysis of that now. We've now got well over 1,000 CRS patients and many hundred controls. The initial analysis looks like it's simply going to recapitulate this study. Is the microbiome the same around the world? Well, Elke addressed this question a few years ago, and he undertook a large international study of more than 400 samples, all of which were sent to Adelaide and analysed in exactly the same way. And he and his team showed that there was tremendous inter-individual variation, but enormous variation around the world, but again, no clear pattern. And nowhere was it markedly different from anywhere else. How about if you look more closely at the microbiome and CRS subtypes? We've just looked at CRS overall versus controls. How about if you look at subtypes? Here is the average microbiome of a group of normal patients. Here is the average microbiome of a group of CRS patients who did not have asthma, the average microbiome of a group of CRS patients who did have asthma, and you can see perhaps there's less Corynebacterium in that group, and the average microbiome of a group of patients with CRS secondary to cystic fibrosis. You can see they're radically different with Pseudomonas, which is rare in the other groups, lots of Fusobacterium, lots of Staphylococcus. But the question to ask here is it indeed the clinical status, the pathogenesis, the pathophysiology of these conditions which is bringing about these changes in microbiome? Or is it in fact the difference in severity of these three different subtypes of CRS, which are being treated by different amounts of antibiotics? Could in fact these changes be secondary to our treatment? Do patients with CRS get a lot of antibiotics? Well, one of my PhD students rather laboriously counted how many tablets of antibiotics in the year prior to operations patients with CRS with and without nasal polyps had, and it was lots. I don't think we're the most parsimonious of antibiotic prescribers in New Zealand, but also I think the idea of antibiotic stewardship is taken reasonably seriously here. Despite all of that, our CRS patients were given a lot of antibiotics. And I suspect a lot of the changes that we specifically saw in the cystic fibrosis patients are because those patients are given very high doses of very potent broad spectrum antibiotics from a young age. And I suspect that that has greatly reduced the community complexity in those patients. So the third question specifically is the microbiome affected by antibiotics and by surgery. And in order to look at this, we wanted to really treat patients and measure the microbiome before a specific treatment and the microbiome after that treatment. And in this study, we had 10 patients who had CRS, they were given doxycycline, 100 milligrams BD for a week, 10 patients who were given prednisone, 30 milligrams for a week, and 6 control patients who were not treated. And here are the average microbiomes for those three groups prior to their treatment, the amount of Corynebacterium, Staphylococcus, Haemophilus, Streptococcus, all sort of similar between the three groups. And after the treatments, after a week of doxycycline, 200 milligrams daily, or prednisone, 30 milligrams daily, not a great difference. Short-term study, others have found different, but you don't see in a single course of antibiotics an enormous change certainly in our hands, in the microbiome. What about surgery? We had 23 patients here before and after comprehensive FES. Here are the microbiomes of the patients at the time of the operation. Here are the microbiomes of the same patients between 3 and 6 months post-operatively. And there was only one consistent change in that we found much more Staphylococcus post-operatively. It was exactly the change we were not expecting to see, given the reality is that the data which supports Staphylococcus having a significant pathogenic role in CRS is far stronger than it is, I think, for any other bacterial species. Most of these patients did well, but they had more Staphylococcus. So this is one of the mysteries. However, having said that, I'd like to talk a little bit more about Staphylococcus and changes post-FES a bit later in this talk. Is the pathogenesis stable over time? This is really important for the sort of studies that I've just been showing you, where you intervene and then see whether the microbiome changes or not. Well, we had 4 volunteers who had their microbiome determined over a 2-year period recurrently. Here's the first, the second, and you can sort of see, here's the third, that although there is a little bit of change over time, that they sort of keep the same characteristics. You could sort of see that one is different from the other, and they are relatively consistent. And that, in fact, is true of the fourth patient who, having said that, in the middle of winter of the first year, developed terrible sinusitis, and at which time her microbiome was overrun with H. flu. She took some Augmentin, and you can see for the next few months, the microbiome was not quite as it was before. And then within a month or two after that, she had reestablished exactly the same pattern she had before that acute illness. So the bottom line is, yes, the microbiome is, in normals, relatively stable over a period of time, but it can be changed with acute infection. How about patients with CRS? Recently, another PhD student has followed CRS patients over a 12-month period. And for the great majority of these patients, during that 12-month, they received sinus surgery. We followed them up specifically so that a sample was taken every month, as well as every time they had an exacerbation, they'd call in and dutifully get sampled. And here's a sort of result that we got from one patient, and you can see that the microbiome changes all the time. One patient, two patients, and so the pattern goes on. Microbiomes in chronic rhinosinusitis patients are extremely unstable. So what we see at one single point in time may in no way reflect what the microbiome will be in a month or 2 or 3 or 6. And finally, I'd like to address the question, does all of these changes that I've described have a plausible role in the pathogenesis of CRS? And here's some preliminary data that intrigued us. And we looked at how much Corynebacterium there was from specimens of CRS patients and how much Staphylococcus there was. And the more Corynebacterium there was, the less Staphylococcus, and vice versa. And this rather implied that there may be some sort of interaction. And this is a sort of suggestion that Peter was making about lactobacillus in the animal model, that there are potentially good bacteria and bad bacteria. And in fact, this is a study that Peter's group published quite recently. And what these data suggest is that there's a bacteria called Dolossogranulin pigrum, and it may in fact be a good bacteria. And what they've found is that this particular bacterium is more prevalent in healthy upper respiratory tracts compared to patients with chronic rhinosinusitis. And then they did co-culture, and they found that the Dolossogranulin pigrum had an antimicrobial effect against Staph aureus. And in co-culture, if that particular bacterium was present, there was less inflammatory cytokines produced by airway epithelium. That if you added IL-4 to the nasal mucosa of laboratory mice, that the IL-4 reduced the integrity of the barrier, but that reduction in integrity of the barrier was not seen if the IL-4 was introduced with Dolossogranulin pigrum. And so the suggestion is that this is a good bacteria. And it may well be that manipulations, and I don't really think we've entirely figured out the best way of doing this, but there may be potential for manipulations in the microbiome which may produce a new stability or a new function of the microbiome, which reduces the severity of the CRS. And I'm going to finish with one final study, which I really like, because it may explain how it is that FES works. Because it's always frankly been a bit of a mystery to me. And what this team did was that they said, well, Staph aureus is a common commensal, and we know that. But what they found was that anaerobes degrade the mucus to form metabolites. And those metabolites encourage the growth of Staphylococcus aureus, but they also encourage the transcription of staph virulence factors. And so their idea was that sinus surgery may work by re-establish aerobic conditions, so you don't have the anaerobes breaking down the mucus, firing up the commensal Staphylococcus. So an idea which I say really appealed to me. So look, I hope I've addressed those questions, albeit briefly. I still don't entirely have an answer whether or exactly how microbiomes or that microbiome causes CRS. But I think there is, to summarize, a great deal of literature, some very compelling arguments that it does, but the complexity of that involvement is frankly far greater than we've originally conceived. And so with that, I would like to thank my team who produced the data which I showed you, and I'd like to thank you very much indeed for your attention. And at that point, Tim, I will stop sharing.
Thank you very much, Richard, for this wonderful presentation, and a lot to talk about, of course. I hope we have some time at the end to discuss. Thanks very much. So the next